My name is Raul Freitas. I speak from the Brazilian Synchrotron Lab. So it's my pleasure to be here. I want to thank the NISPAC for making this possible. I will speak a little bit about our IR program that we have there. As you see, maybe it's a too long title for a 15-minute talk, of course. But let's see. I will try to be fast. So basically, I will not speak that much about the synchrotron technology, but something to remember is that it's a very broadband source, and it has, let's say, a spectral irradiance that's about 1,000 times better than the thermal source. And this is enough to allow there is no experiment to be done with the synchrotron. So this is basically something to remember from this source. And also, this is a picture from our accelerator. This is 40 meters wide. And the good thing to remember also is that having a s-SNOM station in these sort of facilities, you allow people to, even people that doesn't have such instrumentation, to go to this place, to submit a proposal, and actually test this experiment. So this is something important to remember. And for the maybe third or fourth times, you're going to see another time this scheme. But basically, just remember that we take the infrared radiation from this big accelerator. We have usually some optics here to collimate the beam, and then we deliver to the s-SNOM scheme, the interferometer. So this bottom part here is basically the NISPAC instrument in a scheme. Okay, so just to remember, this experiment at synchrotrons, it started in Germany with at PTB, and then ALS also showed results in that sense. And after that, we also reached some results. And now many labs are actually implementing this, which is pretty nice. Talking about sensitivity, specifically in our experiment, we could reproduce a very nice paper from, I think, 2013, from Florian, Reiner, and just trying to have a classical spectrum and try to see if it resembles the classical FTIR experiment. So we can see here a comparison between 120 nanometers PMMA film in the ATR, classical ATR, and the nano-FTIR. But something that we try to learn here is actually how's the sensitivity of the technique using synchrotron. So we start actually analyzing films from some hundreds of nanometers up to 3 nanometers thick films. And we could see actually that we could, let's say, identify the carbonyl peak until 6 nanometers, which is pretty cool. As we know where the peak should come, you know, being very positive, there is something here for the 3 nanometers, but it's already fantastic to see this sensitivity with a very low power source. So if you take the synchrotron source, has few, let's say, microwatts per centimeters inverse. If you compare to laser, of course, it's way lower. So it means that it's a very quiet source. The signal-to-noise ratio is pretty high. So I will speak a little bit of our, let's say, main programs in biology and also in 2D materials. So I start with biology. So this comes from a researcher in my institute. So she was basically trying to understand the color of hair with respect its internal chemistry. So she sliced many kinds of hair, different colors, and she was trying to see actually specifically those, let's say, clusters of melanin, they call melanosomes, and try to see if there are some difference between them. So this is a pretty nice example that you have many peaks in the whole fingerprint range, and she could actually find many of, let's say, expected peaks from hair in these experiments. So this paper has very nice examples of spectra in this kind of applications. You could even see 2 kinds of melanins there and correlate to color. So this is something pretty nice. So another kind of study that people are trying there is to develop nano drugs to, let's say, target those nano drugs to specific living systems. In this case, they are functionalizing silicon dioxide nanoparticles. They put some drugs inside, and then they functionalize in a way that the particles become more, let's say, you increase the affinity between the particles and the system, and you target, in this case, for bacteria. This is E. coli bacteria, and we know that for, let's say, for the case of superbacteria, people are trying to use classical antibiotics. You cannot actually deal with those bacteria, so the idea is to develop something that can be more efficient. So basically, you see the particles here. This is an AFM map, and we took some spectra, of course, on top of the nanoparticle, just the silicon dioxide signal, and then this is the control just with the bacteria, with the two amides here, and we could see in between, when you go from the bacteria to the nanoparticle, you actually have the amide 1 peak being a little bit shifted, so it means that there are some interaction. They were trying to just show that the affinity is actually increasing between the particle and the so I will not speak that much about this, but this is a very cool case. Oksana is going to talk about this later. So basically, we are trying to study isolated brain cells, so neurons, in the perspective of Alzheimer's disease, just to see if there are some differences. Well, this is pretty cool. So finally, this is the last case I will show about biology, and this is a new development there, and it's s-SNOM in liquids. So basically, we are using a platform that we can confine the liquid and also some organelles. In this case, we are testing protein clusters, and we cover this with the graphene. We know that this work here is actually the work that inspired us to do it. Professor Bazov is here. So, and basically, we're trying to do that in a more, let's say, robust way that our community can actually bring their samples and we can try to do it. So we tested 3 systems. Human albumin in water, so we could see clearly the amide peaks. So basically, what we see is that those clusters, they attach to the graphene, and then we can access this dry phase, but it's being inside the liquid that can actually give nutrients to these clusters. And we also tested a salt dissolved in water, and we could see the very nice peak of this salt, monopotassium phosphate, which is pretty important for many biological applications. And finally, we test just a polar solvent to see if we could get the signal from the liquid itself. And we could also detect very nicely here the S-double-O bond here. So this is a work led by Ingrid Barcelos. So she's in my group in Brazil. So now going to the other part of the title, so talking about 2D materials. So first, this is one of the first materials we studied there. We were just trying to understand, let's say, early stages of oxidation of black phosphorus. So basically, we were pinpointing different places for different, let's say, time exposure of these flakes, this 2D black phosphorus in air. And basically, we could see the formation of some phosphorus oxides here. So this was a nice study, but I would say that right now we are more, let's say, looking at these quasi-particles or waves on these materials, these 2D materials. So this is in my group. So it's inspired by many works. Many of people are actually here, and basically, we are trying to see what happens in this classical architecture that people use to tune the graphene, the graphene firm level. And you always have these pads, and we are trying to see actually how light is confined in these systems. So with a broadband source, you have the advantage that you can actually make these line scans, and you have the whole spectrum at once. So we have the 2 Weierstrahlung bands of boron nitride all the time there. So we could reproduce something that's already known. So just boron nitride on top of silicon dioxide, and we could see the dispersion there, and this reflection of these phonon polaritons at the boron nitride boundary. Then we tested on top of gold. Of course, the dielectric environment is different. You have different polariton wavelengths. But then we tested in this, let's say, heterostructure here that's more related to the system when you have pads underneath. And we could see also a sort of reflection here that resembles this. But we didn't know if this is coming from the boundary, the edge of gold, or from the tip. So what we did is we took some hyperspectral image exactly at this boundary, and we had this. We could actually slice this hyperspectral cube in different frequencies, and we take these profiles, and we were trying to fit these profiles. They don't fit actually with a single source from the edge, so we must have something from the tip. So we could actually see that there is definitely a reflection there from this, let's say, virtual dielectric barrier. So I can give more details for you afterwards. But another system that we're testing there, it's actually a new 2D material. This is also I forgot to say this. This work was led by Francisco Maia, which is also a researcher in my group. So this is a new material, what we call TOG. It's a soapstone 2D material. So if you take soapstone, you can actually exfoliate this material, and it's a silicate, it's a insulator, so it could be a good option for device that you cannot use, like say, on-chip silicon dioxide, and you can just transfer flakes. We could see that the material is very flat, it's atomically flat, and it has also some phonon response very close to the range of it's actually the silicon oxide bound. So we put graphene on top of this guy just to see if there are some, first of all, plasmons, and to see if there are some connection between the plasmons and the phonons, and also if we can control those phonons just applying a gate voltage in the graphene. So we could see that actually it's possible to control. So this is a nice example that we can use the synchrotron to study. Okay, so just finishing a few slides here. So we have a machine that's, let's say, it's already considered old, and let's say the form factor of the beam is not so attractive. We need to cut the beam to enter in this microscope. So the point is that there are some room to improve if we actually think about new machines that are coming. So we are constructing a new machine in Brazil, which is, it has, let's say, a different technology for let's say, these accelerators. Basically, it's a more stable machine. And we're going to try to push this range up to the, let's say, terahertz range and explore more this source in a range that the lasers are still not covering continuously. So this is something that we are planning. And this is my last slide. This is data from, let's say, 2 days ago. I just came from Berkeley, and they have there a new detector. Mike is going to talk more about this, but the impressive thing is that we were actually, before, just studying the TOG in this range, or the mid-IR range, and we could see those peaks that I just showed you. But we had some predictions from DFT that actually there are some activity in this range of 300 up to 500 wave numbers, but we had no detector to do it. So then we came to California, and we tested this range here, and it's actually, the signal is there. So it's a nice example of the use of a broadband source that you can, again, have, let's say, the far IR response together with the mid-IR response in a single-shot experiment that you can compare intensities. It's pretty, I think this is the main advantage to highlight. So I want to thank NISPEC for giving this opportunity. This is a picture of my group. This is the instrument inside the box here. Thank you very much.